this year was a continuation of my last year's project, which focused on developing a novel technique to monitor the health of fresh water. I guess I could just jump right into it, give an overview. So my method of utilizing the larval chironomid, which is a non-biting bridge larva in order Diptera. This is an example of what they look like. To monitor the health of fresh water, it adds significant value for managing our increasingly scarce freshwater resources. It increases the accuracy, precision, and as well as statistical power. This method can capture the cumulative effects of habitat alteration and pollutants, things like field sensors and chemical testing cannot. Using macroinvertebrates for bioassessment was something that was popularized in about 1977 by a researcher called Hilsenhoff. There's something called the Hilsenhoff Biotic Index, which is a scale from 0 to 10 on how pollution tolerant an organism is. For example, if you have, show this guy right here. For example, if you have a caddisfly, most caddisflies tend to be, this is a Chironomidae. They have a tolerance value of around 1, which means that they're very sensitive to pollutants. Evolution plays a big role in this, and so do genetics and genomics, which I'll get into in a moment. But I developed a technique, basically a method that has promised to be the world's first global standard for bioassessment. It also has promised to be the first method that utilizes genetics to monitor waterway health, especially a technology called DNA barcoding. So that was kind of more of an overview. This year I focused in on perfecting my method. I compared various methods. Levels of taxonomic identification. So you have order, family, so family Chironomidae. I guess I should also say why I chose them. Chironomidae are extremely versatile. They're free-living, holometabolous, benthic macroinvertebrates. Chironomids are found on every continent, including Antarctica, and can also be found at a great range of altitudes. Because of this, they're a common denominator across global waterways. I discovered this when I was sampling many sites all over Mercer County, and I found that they were present in every site. However, the chironomids from, say, the Millstone River versus Largo Pond versus, Jacobs Creek, they look different. Using some tools, like using a tool called DNA barcoding, in which you extract the DNA and sequence it, I was able to find that they're actually different species. Not only that, you can use the Chironomidae and just the Chironomidae to monitor the health of water and get more accuracy, more precision, and more statistical power than previous methods, which would be manual taxonomic identification by morphology. I looked at statistical power on various levels of identification. I compared identification down to family level. The family biotic index of the Chironomid is 6. However, my research has shown that within family Chironomidae, the genus tolerance values range from 0 to 10. So within this family, you've got the entire range. 10 meaning that they have adapted to withstand very, very high environmental stressors. A good example of how a chironomid has adapted is there is a type of chironomid, the genus of chironomid called the Clinotanipus. The Clinotanipus in subfamily Tanipodidae has evolved to be highly carnivorous, so it feeds on other chironomids. It also is red due to excess hemoglobin in it, which allows it to live in areas with extremely low dissolved oxygen. So that's just an example of how there are so many niche species of Chironomidae. Is this making any sense? I'm not sure. The second phase of my project was applying it to real world. I did a microanalysis along different sections of the Stony Brook using my method to track non-point source pollutants. I guess I can't really show this part, but and using GIS software to plot it as well. And the third and possibly the most important section of my project was establishing real-world application in the local watershed institute in Pennington, New Jersey. I now have a genetics lab fully up and running. It took a couple years to get it in, So the third part of my project was comparing barcoding macroinvertebrates within that lab, which can be used to implement this new procedure, and also be used to teach high school groups basic genetics, identification of threatened and endangered species, which all can be used to enforce environmental regulations to protect our remaining clean bodies of water, and also to protect threatened and endangered species as well. I compared, I got some samples from the Arctic Circle in Norway, some amphipod samples, and I compared, I cut them into sections, and I compared identifying them in a professional research laboratory, or Cold Spring Harbor Laboratory in Long Island, New York, to identification at the Watershed Institute. And the results proved identical, even though I was using the different equipment, different procedure, and in-house made reagents. So that showed that that was kind of like a proof of concept that this lab was functioning. Additionally, although this is less scientific, but to get the lab on its feet, it required a lot of writing grant proposals, standard operating procedures, training staff, and a lot of trial and error. I also did another test with a known species as a control, Phaeocetta domesticus, or the domestic house cricket, which is commonly used as a feeder animal for various amphibians and reptiles, and captive animals that would otherwise feed on insects. And these crickets were cut in different sections, also barcoded as well, and all of them worked, and all of them sequenced correctly as the corresponding species.